Meow. All right. Do, do, do. All right, good morning. Preparing oneself for the onslaught of meiosis. I hope you're all fully limbered and prepared. Make sure I got my markers. All are present. All right, I'm going to get cracking because we need to make hay, so to speak. Right, so last time we finished off a little ways through the phases of mitosis, I believe. Right, and so we really need to finish that off before we can get on to meiosis. So I'm going to just whip through that quickly. I believe that we finished up here, right? Metaphase, talking about the M phase checkpoint. Anybody jog my memory? Wednesday was a long time ago. Yeah, okay. So essentially we talked about how cohesin was holding together the sister chromatids. We'll get to that in meiosis. It's a very important point. And how Shigoshin protects the cohesin around the centromere until all these chromosomes are lined up, right? So essentially, once that happens, Shigoshin gets destroyed or kind of gives up the ghost, essentially. And the cohesin is, destroyed by another protein called separase. And because essentially there's tension, right? You can imagine I'm trying to pull my hands apart. And cohesin is the only thing keeping those two chromatids together. Ah, no. I was having this problem yesterday. I don't know. You can hear? Oh, gelling, that's a relief. Okay, cool. Okay, whew, phew, that was a relief. I had a little panic attack there because I had problems with WebEx yesterday, although that program's a complete turd. Okay, so, all right, so anyway, we've got these chromatids and they're trying to pull my hands apart, right? And essentially separase cleaves or destroys the glue. And so that tension is still occurring and they just pull apart, right? That's really what anaphase is, right? And essentially that then morphs into telophase and cytokinesis, which kind of occur at the same time. They're not really distinct phases. And so the cell kind of goes from round during metaphase and it extends in anaphase. And where my fingers meet were where the chromosomes were aligned, that metaphase plate. That now serves as a kind of like a ring, right? There's actually proteins that are deposited there, pardon me, which kind of help pull in and contract, right? That bit between those two new future cells, right? That's called the cleavage furrow. In animal cells, at least, plant cells are a little bit different. And at the same time, those chromatids or the chromosomes now, right? Because they're separate. Those chromosomes decondense, right? Remember what I said about telophase being the opposite of prophase, prometaphase? Essentially, this is what's happening, right? The nuclear membrane reforms, the spindle fibers disappear, and the middle gets pinched off. And it looks pretty much like this, right? So there's this contractile ring, which very much like if you're trying to pinch a balloon in half and then like twist it, if you're a clown maybe. That's exactly what's happening. That kind of ring is getting smaller and smaller and smaller and smaller, pink, until it pinches off the cytoplasm, cell membrane kind of separates, and now you have two daughter cells. The only difference in plants is that, you know, they don't have flexible cells because they have a cell wall. And so instead what we have is a cell plate form, which you can see here, where these are basically small compartments of cell wall material. And then you end up having that new rigid cell wall between them. Okay, there's a bit of a quick roundup because we want to get on to meiosis. Anyone have any questions about mitosis before we move on? Nope. All right, awesome sauce. No, that was mitosis. We want this one. Get rid of you. So I want to get across the big picture quickly, again, because I'm pretty cognizant of time here. So the big picture of meiosis is random generation of variation. So meiosis is one of two parts of sexual reproduction, the other one being fertilization, right? And both meiosis and fertilization, the aim is to essentially produce a random combination of alleles in the new generation. So that's a random combination of your alleles as an individual, a random combination of your partner's alleles as an individual, and then randomly picking which of those two random combinations come together to form a zygote, and that forms the next generation, essentially, right? And so the reason for that, and sex is costly, right? In terms of inheritance, you only pass on half of your genetic material to your offspring, right? So, you know, if you want to, it's all about me, right? Then asexual reproduction would be the way to go. So there has to, and also, you know, just the act of reproduction can be pretty costly, ranging from, you know, black widow spiders where you get eaten because, you know, go next generation. So you give your resources to the next generation, essentially, which is very selfless, I guess. To even things like snow leopards, right, where, you know, the mating male often tries to kill the pups of the previous year's litter, right? So sex is costly in a whole bunch of different ways, right? So it must have some benefit. And the benefit is that random generation of novelty, of not novelty really, because you're working with what you have, right? But essentially reshuffling the deck, right? And the reason for that is because the environment in which we exist is not static, right? And we're sufficiently complex as organisms. I'm looking out the window because I'm like, ooh, it's cold out there, right? Clearly not a static environment. It was only a few days and it was almost t-shirt weather, right? For non-San Antonians. So the environment is static and you have no idea what that environment is going to be for the next generation, right? As you're in the process of producing it. So sexual reproduction and, you know, the randomness of meiosis and fertilization basically allows you to put out offspring which have different combination and potentially higher fitness if that environment changes, right? Obviously, there may be some that have lower fitness, but if you're talking about wanting your next generation to survive, then it will do, right? If it's all the same as if you were reproducing asexually and say suddenly a new predator arrives or water dries up or whatever, then you're kind of screwed, right? Or your offspring are. And so that's really like the purpose of sexual reproduction. And meiosis is a big part of that random generation, that random reshuffling, right? And we'll talk about further. So there's really three ways. I'm going to write these down. In sexual reproduction, right? First way, and I'm going to start kind of the end and work backwards in a sense, is the act of fertilization, which by large part, and there are a few genetic modifiers of this, sperm do have different fitness. And, there are obviously behavioral modifications to reproduction, which change the odds of one individual succeeding over another. But generally speaking, the act of fertilization is random, right? So which oocyte a female will generate out of the pool of oocytes that she has, and which sperm out of potentially millions of sperm produced by the male will succeed in fertilizing that oocyte is random, right? So that's a giant, kind of mixing pot essentially, right? Second way is another big one, right? So this is the random alignment of homologous pairs in metaphase 1. Right, and so essentially, if you remember from mitosis, right, you have your this is one homologous chromosome, this will be another one. These are chromatids, right? 2 fingers essentially in each hand, right? They line up like so on the metaphase plate, right? They kind of jiggle around like so until anaphase when they pull apart. Well, in meiosis, those homologous chromosomes are paired up like so, right? They're actually held together by what are called chiasmata, which we'll talk about towards the end of the lecture, right? The act of recombination. So they're actually kind of like that, like so, kind of. Ha. Right. Gets a little bit difficult because there's only so many ways I can bend my fingers, right? However, that aside, we'll get to that in a bit, right? It is completely random, like how, like these are kind of stuck together, right? If you imagine this is in the, pro-metaphase kind of thing, right? So as those spindle fibers get shot out by the microtubule organizing centers, the MTOCs, right? It's random which side they'll hit, which one of these pairs, right? If the one at the bottom hits this one, then this will get pulled around until it's on the metaphase plate like so, right? If it's randomly that the spindle fiber from the bottom hits this one, it'll be like so, right? It's completely random which way round it is. 50-50 chance for every single chromosome pair, right? So if we have 50 chromosome pairs, you get 2 to the power of combinations, right? So this is like the potential number of different combinations, right? Is it going to be chromosome 1? It might be like this or like this. Chromosome 2 might be like this, might be like this, right? For every single chromosome pair. And we have 23, right? So every time a human cell goes through meiosis, we have 2 to the power of 23 different potential combinations of, maternal versus paternal chromosomes, right? the one that you got from your mother, the one that you got from your father, right? And that is 8.4 million different combinations. Obviously, you only get 4 cells from one round of meiosis. So essentially, you have each one of those is what, one in about 2 million different potential combinations, more or less, right? So that's essentially this. The analogy I like to use is like, essentially you have 2 decks of cards, right? One that you got from one that I got from my mother, one that I got from my father. And so during meiosis, this random alignment of homologous pairs is essentially you're just shuffling those cards, right? For this pack, I'm going to get ace of spades from this pack and get ace of spades from this pack, kind of doing that essentially. You're just reshuffling. So at the end, you're going to get a whole new deck of cards, right? Which has some cards from one parent, some cards from the other one. Which ones you get is random. This is also for the basis for the law of independent assortment, right? So this is the actual cytological basis for Mendel's law of independent assortment, which applies to dihybrid, trihybrid, whatever crosses, wherever you have 2 or more genes, right? We're assuming that they're on different chromosomes. So, for example, here we go. Both of these are heterozygous for 2 different genes. They're on 2 different chromosomes. And depending on which way round they are, you get these different allele combinations in the gametes, right? But because you have equal probability of these, essentially you get a 1:1:1:1 ratio of allele combinations in the gametes. You think back to a dihybrid cross, how many different gamete combinations do you have in the produced by the F1 generation? 4, right? What ratio are they in? 1:1:1:1, right? And that's what comes together to form the F2 generation, right? So this is the actual basis of the law of independent assortment. Any questions so far? All right, and do ask if you have them, right? Because this is kind of the easiest point at which you can ask those questions, because you have the exam on Monday. Okay, and we can talk a little bit about that at the end of class, if anyone has any questions. Okay, so the third way of generating new combinations, right, is recombination. I'll move this over here for a sec. Between non-sister chromatids of homologous chromosomes. Right, and what that really means, like in kind of layman's terms, essentially, is we swap bits, pardon me, of homologous chromosomes. And that basically generates allele combinations that are not present in the parental chromosomes. Right, and so stretching the 2 decks of cards analogy further, that would be like, you have 2 decks of cards and they differ slightly in how kind of dirty or rumpled or people doodled on the back of them or something like that. And in recombination, essentially what you're doing is you're getting 2 aces of spades. and you tear off a little bit from one of them, and you tear off the matching part from the other one, and you just tape them onto the opposite cards, right? That's really essentially what recombination is about. And so now you have a card which is not was not present in the original deck. Okay. Questions. We'll go through both of those in a wee bit. I don't know why people always use our driveway to turn around in this place. Anyway. Okay, so some big points about meiosis, right? It's production of gametes, sex cells, right? Those gametes are always haploid in sexual reproduction. And the basic reason is the only way you can get 2 is add in 1 and 1, right? So we're diploid, we have 2 copies of each chromosome, 46 total for all of us. And the only way that you can get 2 copies of each if you start with 2 cells which originally have 1 copy of each. So 1 + 1, 2. Simple as that. Right, so meiosis, aside from all that other jazz that we're talking about, its other main role is to generate those haploid cells, those gametes, right? And so to do that, we have 2 rounds of cell division, right? We have meiosis 1, which is called reductional division. That's actually where all of the really important stuff happens, right? So meiosis 1 is where we go from diploid to haploid. Also called reductional division or reduction division, right? Because you're reducing the chromosome count. Well, really the ploidy is probably the better word. And also, whenever you talk about meiosis, you have to be very specific about which one you're talking about. So you can't say metaphase meiosis because metaphase 1 and metaphase 2 are very, very different, right? You have to be very specific. And when I ask you questions about this stuff, I will also be specific because I'm going to be asking you something very particular, right? So meiosis 2 is haploid to haploid. So this is really just separation of sister chromatids. Again, that's called equational division, right? In another key point, mitosis, 1 cell, 2 daughter cells. Those daughter cells are identical. Meiosis, 1 cell, 4 daughter cells, non-identical. They are definitely genetically different, right? And also part of the reason that they're genetically different is both the random alignment of chromosomes, homologous pairs, and also the crossing over or recombination. Right. So this is a kind of pictorial description of basically what I just said. Right. We have interphase kind of over there, right? All the preparation. And in meiosis 1, we go from 2 of each chromosome to 1 of each. Which chromosome each daughter gets is random, hence the random alignment of chromosomes. And then meiosis 2, we separate those chromatids. This is not the full picture because we have recombination occurring in meiosis 1. And so you will not get this. This is a simplification. We're just illustrating the reductional versus equational division. Okay, so essentially we have 2 rounds of cell division, right? So we have the same series of phases that we do in mitosis. However, in meiosis 1, we have a lot of funky stuff going on here, right? So, sorry, and some allergy issues, right? So this is all prophase. So we have prophase, metaphase, anaphase, telophase. And really in kind of in here, there's prometaphase in between as well, right? But what we really care about in meiosis 1 is this prophase part, right? And I want to draw out what's going on here, right? So just in kind of general, you have the same things going on as in mitosis, right? You have the centrosomes moving, you have the chromosomes condensing, you have the nuclear membrane disappearing, spindle fibers forming and attaching to chromosomes, chromosomes lining up in metaphase, chromosomes coming apart in anaphase, right? Not chromatids, and then telophase, potentially, depending on the organism, right? So that seems similar to mitosis, but there's some very, very important differences. The main one is that, one, we have recombination occurring here, which we'll talk about in a second. And two, that means that chromosome pairs line up, not individual chromosomes. Chromosome pairs line up on the metaphase plate. So you can see that here. So if you ever see a picture, hint, hint, wink, wink, in an exam of a cell, and you have chromosomes in, these are called tetrads, right? Because there's four individual bits stuck together. If you see this, that's metaphase 1. Instantly tells you without a shadow of a doubt that you're looking at meiosis and metaphase 1, not mitosis, not meiosis 2, right? Now that those then separate and then we get the telophase, right? So I want to draw out what's actually happening here because this is super, super important for a bunch of different reasons. And as I try and kind of tell you at the beginning of the course, I tend to bump around a lot in these PowerPoints because it kind of depends on how my train of thought at the time and what I need to emphasize. I'm not necessarily going to go through these linearly, right? So I'm going to jump around to talk about recombination now. And I want to do that with a diagram. Okay. Get to the back of my couch again. Isn't that lovely? All right. I'm going to close my curtains. Okay, that's it. All right, so prophase 1 equals recombination. Okay, trying to make sure you can see as much of it as possible. All right, so, yeah, good point. Thank you about that. Trials and tribulations of online teaching, right? Okay, so recombination, it's important for 2 reasons. You get to see my fingers there, right? First reason, it generates that genetic novelty. Not novelty, variation. Novelty implies we're generating something new and not. We're generating new combinations of the same things. The second reason that's really important is that these recombination events, right? essentially, when we're crossing over here, this is what holds the chromosome pairs together until anaphase 1. This is why those chromosomes stick together all the way through late prophase and metaphase until they get pulled apart in anaphase, right? So there's really two really important reasons for recombination, right? So I'm going to just draw up two different this is a pair of homologous chromosomes, right? So this is one homologous chromosome. Here we go. One homologous chromosome, another homologous chromosome, right? This makes our pair. These two are identical sister chromatids. These two are also identical sister chromatids. This chromatid and this chromatid from the different chromosomes are not identical, right? Even though they have the same genes, they don't necessarily have the same alleles, right? So let's say we have, let's tip this down a little bit. I'm writing them at the side simply because it gives me a little bit more space, right? So during early prophase, these chromosomes are perfectly paired up next to each other, right? Base by base. It's not like that. It's not like that. It's exactly aligned, right? And they're also pardon me. Wish I had another color actually. Never mind. These chromatids are held together by cohesin. And that cohesin is protected at the centromeres, right, those dots, by, can anybody remember the funky Japanese word for the thing that protects cohesin? Yeah, Shigoshin. This is super, I think it's Shin or Sin, one of the two. Can't remember right now. Right. This is going to be really, really, really important because this is what controls how these two or these four different bits pull apart. Shigoshin is what basically allows the homologous pair to pull apart in anaphase 1 and then gets broken down later, much the same as in mitosis, to allow the sister chromatids to separate in anaphase 2. Right, but we're not going to go quite there yet. Right, so we're going to have a crossover form just here. Right, can you all see that okay? I'll post the picture up to Blackboard after class as well. Okay, so again, we've got a homologous pair of chromosomes. This homologous chromosome has dominant alleles, and it's AA and BB. Those are the sister chromatids, right? It's not that this is homozygous dominant, this is homozygous recessive. It's just that there's a allele here, and because this one's identical, there's an allele here, right? Just to be clear, don't want to confuse things. Right, so we're going to have a crossover event between these two genes, right? All right, so what happens now, and we got like this little progression going on. is that we get a break happening just here, right? So we're actually, oh, it is Shin. Oh, I thought it was, but it doesn't really matter at the end of the day, right? So we get one of those, each of those chromatids, and these are non-sister chromatids, right? These are the ones next to each other. This is really important because that means that the ones on the outside will be unchanged, right? So you'll always get a parental, recombinant, parental chromosome from this crossover, right? Now, this strand then gets attached to that one, and this strand gets attached to that one. Now, all the while, these sister chromatids are stuck to each other with cohesin. So now, when we have spindle fibers, let's draw the centromere here, pulling on this pair, right? This will be in essentially pro-metaphase 1, right? Between late prophase 1 and metaphase 1. Now we have this chromosome, and let's actually just draw up the alleles again, so I don't forget, because this is also important. I'm essentially teaching you two different things at the same time. So hopefully I don't lose you in the process. Right, so we're trying to pull this pair apart, right? Like that with our spindle fibers. However, this chromatid here on the outside, right? Not on the outside, inside, sorry. This chromatid here on the inside is physically attached to this chromatid, right? They're continuous now. And this chromatid is attached to the one on the outside with cohesin. Likewise, this chromosome here, this chromatid is physically part of this one, right? Because it's swapped over. And this chromatid is attached to its kind of sister, I guess, now, not quite the same, with cohesin. So cohesin here is what holds the homologous pair together up to and including metaphase 1. Does that make sense? Right? It's not the cohesin down here by the centromeres, right, which is protected by Shigoshin. That doesn't really do a whole bunch in holding the homologous pair together. It's the cohesin at the ends of the arms of the chromosome that hold the homologous pair together as a result of this crossover. So this is, got a little bit of space here, called a chiasma. Oops. Or a crossover, or a combination site, any of those words does the job. Okay, so now we've got these all aligned in the metaphase plate, right? We have all of our homologous chromosomes under equal tension. They're all aligned, right? So we get the go-ahead for separation, right? So separase comes in. Now, the Shigoshin down here protects the cohesin around the centromeres. It does not protect the cohesin at the arms. So what now happens is, if we move one step further along, is because that cohesin, right? Remember that the cohesin was keeping this red bit attached to this chromatid over here. That is now gone. Oops, need the green one. Right, so we still have cohesin here protected by a Shigoshin. And the same for our other one. Now, let's go and have a look back at the alleles, right? So remember, at the very beginning, we have one homologous chromosome that has dominant A and dominant B. The other chromosome is recessive A, recessive B. Actually, I'm just gonna put these a little bit closer together, give myself some space. Right. Now we have dominant A, dominant B, right? That's the same as what we had in the parent. We have recessive A, recessive B. Yep. However, we now have another mix. We have A, recessive A, dominant B, dominant A, recessive B. So now we have what are called recombinant chromatids, right? These are the parental ones. They're exactly the same as the ones over here. These ones in the middle are now recombinant, right? They're a different combination. Now, if we were to squeeze this in here, right? So now if we go through into meiosis 1, right? So these are essentially going to be in separate cells. Now we're going to end up with the Shigoshin being destroyed, cohesin disappears, and now we have this chromosome, we have this chromosome, we have this one. Ran out of space a little bit, I'm afraid. We have 4 different chromosomes with 4 different combinations of alleles. Sorry about the lack of space, right? So this is what you get. Essentially, this is prophase 1. This will be metaphase 1. This will be anaphase 1. And this will be the end of telophase 2. Right? So their line, crossover, prophase 1. Well, the chromosomes are aligned to each other in prophase 1. They line up on the metaphase plate as a homologous pair held together by this chiasma in metaphase 1. When those chromosomes are pulled apart in anaphase 1, we end up with a chromosome that has 1 parental chromatid and 1 recombinant chromatid. The same for the other one. Right now in meiosis 2, it's all about separating these 2 chromatids. So at the end of meiosis 2, we get these 2. And for this chromosome, we get these 2 chromatids. Questions? So a lot to take in, I know. And I'm going through it pretty fast as well. Oh, generally. That's a fabulous question. The easy answer is I don't know. The more complex answer is something called the anaphase promoting complex, right? Which is a bunch of proteins around that kind of associate with the kinetochore. Those sense tension, mechanical tension. And as soon as all of those chromosomes, all those kinetochores sense equal tension, not here, obviously, over here in either mitosis or in metaphase 2. And then I've read about this and I don't fully understand it, but there's a whole bunch of what are called kinases, right, which are proteins that modify other proteins. And there's a cascade where one protein will modify another one, which will modify another one. And again, I don't fully know whether I've read about it, but I don't fully understand it, whether Shugoshin is destroyed or if it's inactivated. But essentially, the end result is that the Shugoshin is no longer kind of covering up cohesin, and the enzyme separase is able to destroy cohesin. The precise mechanism, I think it is known, but it's not very easy to understand. There's an awful lot of stuff going on. So not a very good answer, I'm afraid. But basically, there's a whole chain of events which lead from equal tension on all the chromosomes. And that sets off a cascade of processes which then modify Shugoshin in some way, which removes it from cohesin. So, yeah, but like honestly, I've read about it for years and I still don't like fully understand that process, to be honest. It is pretty crazy. I actually work on some of the components of that in my own research, but in a different context. And so every time I read about it, I'm like, oh God, don't really understand this very well. Okay, so what's left? We've got 5 minutes. Briefly touching on interkinesis, right? So meiosis is different from mitosis in that there's no interphase, technically speaking, between meiosis 1 and meiosis 2, right? So almost all of the preparation for both rounds of DNA division and cell division have already been done before prophase 1, right? There is a little bit of stuff that goes on between them, and it depends on what organism you're looking at. But the main thing is that you need to make more centrosomes, right? That's the main deal between meiosis 1 and 2. Okay, we don't want to use that. This is basically a summary of the stuff that we just covered in terms of like the molecules involved in those different phases. Again, I really want to emphasize that cohesin is different whether it's on the arms or around the centromeres. The difference is Shugoshin, right? And so that's what allows you to hold your homologous pairs by the ends, right? That and the crossover, right? So essentially this finger stuck with this one and that one stuck with this one, right? And so that differential destruction of cohesin, right? Basically in anaphase 1, we destroy it at my fingertips, pulling it apart. And in anaphase 2, we destroy it around here, which I'm not going to do, and pull those chromatids apart. That's what allows that stepwise division of DNA from pulling apart the homologous chromosomes. So we get a reductional division from diploid to haploid. And then in meiosis 2, pulling apart. So these are now only held together by the cohesin at the centromeres, which is protected by Shigoshin. Now destroy that Shigoshin, destroy the cohesin, and now you get your separate chromatids. Right, and that is illustrated really nicely here. And also this is a good comparison between mitosis and meiosis, right? The differences between the two. And there's also a kind of comparison here as well. So really in meiosis, almost all of the interesting stuff occurs in meiosis 1, right? The random alignment of chromosomes, the recombination between chromosomes to generate this, get down here, these 4 daughter cells, which are all different from each other, right? And so here you can see the bottom left and the top right daughter cells each have parental chromosomes, and they actually have the opposite of each other, obviously. Top left and the bottom right each have recombinant chromosomes, right? So these are chromosomes that you do not see in the parents. This is random. So if one were to do this again, so a second round of meiosis with another cell, you will get a different combination. You might get parental and recombinant, parental, recombinant, parental, recombinant, parental, recombinant, rather than parental, parental, and parental, parental, for example. You will also get a different recombinant chromosome 2, or chromosome as well. Where do we at? We don't have a good picture of recombination here. This will do, right? Because where this crossover occurs, right, varies. And it's random too, right? So we could have this crossover occur here. It could be here, 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 anywhere. It could even be up here. In which case, you wouldn't see any difference in combination of these 2 alleles, because the crossover event would be outside of those. The crossover event has to be between them to see a different combination. There'll be one down here. Could be here, could be here, could be here, right? Where crossing over occurs is also random. So every time you do go through meiosis, right, to get these 4 non-identical daughter cells, you're going to get 4 different non-identical daughter cells every single time. Okay, and that's pretty much it for class, I'm afraid. I'm sorry that was a bit of a wild ride. It was just by necessity, unfortunately. However, I will be available over the weekend if you need questions answered, or a quick meet to go over something that's sticking that doesn't make sense on your own. So please do take advantage of that. The exam will be available from 9 a.m. on Monday until midnight on Monday. Right, you will have an hour and 15 minutes to take it. Right, so, and it will be, I think, 40 questions. Right, so you need to know your stuff, because that's not a lot of time for 40 questions, and that's part of the exam process, right? I'm not trying to be, douchebag. It's actually a way of testing how well you know this stuff, okay? However, you can take that exam at any time during that time period from 9 a.m. to midnight. Also, I will be present in this Zoom at 9 a.m. Yeah, probably till 9:45 or thereabouts. Maybe 9:30 if no one shows up. But for at least half an hour, if not a little bit longer, just in case anyone has any problems with the exam, right? After that, I will be around during the day. Yeah, this is this coming Monday. Where's the schedule? Is it that one? No. Shit bits. Is it that one? Yeah. Yeah, 15th, JM. It's all in the schedule. That's when your first exam will be. I think you're closing a house or closing a store. Work. Okay. So again, you have the whole day from 9 a.m. to midnight. So hopefully there should be a time during that day when you can have the opportunity to sit down somewhere with good internet and take the exam. Yeah, but I'll also be available throughout the day as well if you do have any questions. I just can't guarantee that I'll be around, uh, within like a half-hour time period, but I will make sure I check my email throughout the day in case anyone has any questions. I think that's pretty much it. So study hard, practice everything that I've told you to practice, and good luck for Monday. And I may see some of you on Monday. If not, I'll see you all on Wednesday. All right. Have a fun day, everybody. Good weekend too.